In the earlier video, we were looking at the relationship between DNA, RNAs, and amino acids, and how the cell uses all these molecules in order to make a polypeptide. And that process is known as protein synthesis, right? So, but what we have to understand here is protein synthesis is actually split into two different processes. The first process of protein synthesis is transcription, where the gene is transcribed or copied to produce the mRNA. And the second part of protein synthesis is known as translation, where the mRNA goes to the ribosome and then the tRNA carries the amino acids to the ribosome and a polypeptide is synthesized. So in this video, we are going to be looking at transcription first. So remember, transcription is just when the gene is transcribed or copied to produce the mRNA. You see, in the process of transcription, what we have to understand first is the gene has to be copied to produce mRNA. Now, when I circle the gene, which is found in the DNA, the gene is made up of, well, it's basically DNA, right? And it's double-stranded. But the mRNA is single-stranded. So here's where it becomes a bit peculiar. How is it that something double-stranded produces something single-stranded, which is the mRNA? So we have to understand that process of transcription in detail. So without wasting any time, when transcription happens, only the section of the gene unwinds. Unlike DNA replication, where the entire DNA has to unwind, right? But in transcription, only that particular section where I've highlighted has to unwind, and the bases are then exposed. Now, for transcription, only one strand, which I'm referring to as the template strand, is required. It does not involve both the strands. So I've labeled the template strand. I've also labeled the non-template strand. Template meaning to say it's required for transcription. Non-template meaning to say it is not going to be required for transcription, like that. Now, I'm going to zoom in and only on the template strand and put out a base sequence. So the base sequence of the gene in the template strand there can be many different types of base sequences. You don't have to memorize the base sequence, by the way. Some of my students are like, oh my God, do I have to do this? Do I have to memorize the entire sequence? No, that's ridiculous, okay? Base sequences are not to be memorized. I mean, unless you're a masochist, then go for it. But you don't have to. So I'm drawing out a base sequence over here. As you can see, a TAC, GGC, TTC, AAG, CGC, GCT, and ACT. All right. When the template strands when the base sequences are exposed, what needs to happen is RNA nucleotides will go towards the template strands and form complementary base pairings with them. So remember, when RNA nucleotides form complementary base sequences, RNA nucleotide A will pair up with the template T, but if the template has a base known as A, the RNA nucleotide has to be uracil or U, because remember, RNA nucleotides do not have thymine, they have uracil instead. So if the DNA has a base sequence of TAC, the RNA nucleotide sequences will have to be AUG. That's how it works. And they form the hydrogen bonds between each other. That's what happens like that. And I'm just writing out all the RNA nucleotides so you can just compare and see if I did it correctly. All right. So what happens then is they form hydrogen bonds between the RNA nucleotides and the template strands. And once that happens, another enzyme comes along. And once that happens, an enzyme comes along. And the name of this enzyme is called RNA polymerase. Remember, in DNA replication, we had to use an enzyme called DNA polymerase to join DNA nucleotides together. So obviously, if you want to join RNA nucleotides together, you have to use something called RNA polymerase. It's in the name. It polymerizes or joins together RNA nucleotides. So the RNA polymerase will basically move along the nucleotides, and as they move, they join the RNA nucleotides from a 5' to 3' direction. This is how you get the mRNA, and the mRNA detaches from the template strand, and that's how you get the mRNA, basically. Okay? And what then happens to the DNA? The DNA just basically reforms and it winds back up together. That's basically the process of transcription in detail.